So always transport across the membrane, transport through the phospholipid bilayer. Passive, no ATP required because the molecules are moving with the concentration gradient. So quite the same, but I've got the they're moving from where there's a higher concentration of whatever molecule it is toward where there's a lower concentration of the same molecule, right? Because they're moving with the concentration gradient, or people say down the concentration gradient, that's why there's no ATP required. The types of passive transport, we talk about diffusion, and sometimes people will label that as simple diffusion. Okay, it's the same thing, simple or just diffusion. Facilitated diffusion. And the last one is osmosis. So let me just real briefly define this. So diffusion is diffusion of any molecule. In cells, this is going to primarily be pretty much the only things that can move by diffusion are gas, okay, by simple diffusion. So this is like oxygen, carbon dioxide. Facilitated diffusion, you need a protein to help that molecule move across the membrane. And it's usually because the molecule is charged or it's kind of bulky. So it needs help getting across the membrane. But that still doesn't require any ATP. Osmosis is defined as the diffusion of water. If you can remember that definition, that will help you remember how osmosis works, okay? Some people, and this is again one of those things where there's two ways to look at it, and you can memorize it one way and puzzle out the other way, or you can try to memorize both and not get them mixed up. I personally find it's easiest to remember just one thing, and then if I'm asked the opposite question, to sort of figure it out as I go along. But you're welcome to do whatever is easy for you. So for osmosis, I like to define it and be sure that people are clear. It's diffusion of water, so I like to think of it as the movement of the water. And I understand that perhaps in biology, in high school or AP Bio, you were taught that osmosis is all about the water following the salt, because inside cells, like inside your kidneys, that's how the water is going to move, right? It's going to move to where there's a higher salt concentration. And it's the same, we're saying the same thing. It's just, I prefer to talk about it as just the movement of the water, because in some cells, the salt can't diffuse. I should say in pretty much most cells, salt or salt ions can't diffuse by themselves. They only move by facilitated diffusion, and it's really complicated. You need to go to cell biology in graduate school to learn all about how that works, okay? So I would like to keep it super simple and just focus on that diffusion of water. So this is where we come up with those different types of solutions that are hyper, hypo, and isotonic. Now, those words describe a solution. They describe the concentration differences between a solution and a cell, in the solution inside the cell. So I'm going to jump over to the other side here and see if my blue works. So I'm going to draw those three beakers for this discussion, because this is how it always looks in the textbook, right? So these are supposed to be beakers, and in the beakers are a solution. So I'm going to draw that in purple. This build is blue. Right, so I'm going to make the solution blue for water, right? So the solution, one solution in the beaker has water in it plus some solutes. Right? So remember, water in a solution, water is usually the solvent. Okay? So I've got three different solutions here, and they're going to have different amounts of solutes in them, different amounts of dissolved things in them. Now, I guess it's close enough to Halloween, I can start to use orange, right? So here's my cell. That's supposed to be a cell. And inside the cell, as we learned last chapter, most of what's inside the cytoplasm, cells have a pretty high concentration of water. I know that's very small and you can't see it very well, but you just have to take my word for it. And we usually say between 80 and 90% water inside the cell. So 80 to 90% water inside the cell. That definitely won't fit in there, right? So now, if I put that cell in a solution that is completely empty of solute, if this solution is 100% water, meaning nothing else is dissolved in it except those few cells that I put into the solution, those words, hypertonic, hypotonic, isotonic, they can describe either the solution outside the cell or inside the cell. I'm going to always refer to them as the solution outside the cell, the solution in the beaker, okay? So any solution that has 100% water outside the cell compared to 80% water inside the cell, I'll give you a minute to think about it. But remember, we said it's diffusion of water, and it's going to be from where there's a higher concentration of water to where there's a lower concentration of water, because this does not require any energy or any ATP. So not volume, concentration of. So there is 100% water outside the cell and only 80% water inside the cell. So you always have to ask yourself, where is there a higher concentration of water, inside the cell or outside the cell? In this case, outside the cell, so the water is going to move into the cell. So let's think about what's going to happen to our cell first. If water is going to move into the cell will swell. If that concentration gradient is severe enough, it will lyse or burst. That rarely happens though, because cells have mechanisms to control this, okay? But in theory, if this concentration gradient was enough, the cell would burst and then it would be dead, obviously, right? This solution, where we have a higher concentration of water outside the cell than inside the cell, is hypotonic. Hypo. And myself, that's all that I study and remember. Because, again, I like to remember one thing and then just apply it to the other thing, right? It's over closer so you can get a little bit better. I know it's not great, but it's the best I can do. So I like to remember, okay, hypotonic, 100% water, high concentration of water, my cell is going to swell like a fish, right? Now I can figure out the other two. I can figure out the exact opposite is hypertonic, right? And I'm going to put that in the far beaker for a reason, because I'm going to put the one that's isotonic in the middle. So hypertonic then has to be the opposite situation that I've drawn in my hypotonic beaker. So rather than having 100% water inside the solution, this time I still have my same cell. So I still have like 80% water inside my cell. However, this time I'm going to put, let's say, that's dying already. I'm going to put like, let's say 75%. 75% water in this beaker. So what does that mean? That means it has other stuff in it. It has other solutes in it. So in addition to the water, so 75% water, there has to be, I'm going to put in some salt. Let's put in 10% salt because that's easy math for me. And then 5%, so that's 85, 5% amino acids, and let's say 5% glucose. Right, 10, 20, oh, and 5% something else. I don't know, phosphate. Okay. So there's all of these are solutes in this solution. So there's water in here, but there's all this stuff. And what I need you to focus on is if there's 75% water outside the cell and 80% water inside the cell, now we have to look at where is there a higher concentration of water. Higher concentration is going to be inside the cell, so the water is going to move this time from inside the cell into the solution. It's going to diffuse. It just does. Right? It's just kind of one of those natural laws. Like I said, it's a chemistry thing. Now my cell is going to do the opposite of swell. It will shrink. And if it's a red blood cell, this has a special name, not important. Go to A&P, learn all about it. What you need to know is the cell is not in equilibrium, and so the cell is going to shrink and it won't be able to do its functions properly. And it could die, of course. It's a plant cell, that's why they wilt, right? It's like, if I lean over, it's because there's not enough water in all their cells to hold them upright. If you can remember those two, like I said, I would just remember this one. And then if I ask you a question about this one, you've seen it, you've studied it, you know what the definition is. It's the opposite, it's the exact opposite. So we call this solution of water here hypertonic solution, this solution. Inside the cell is hypotonic relative to the solution. That's not important for me in Bio 1 right now. If you can tell me about the different kinds of solutions, I would be doing a tap dance. That'd be lovely. Although I don't know how to tap dance, so. Emmanuel, do you have a question? There we go. Oh, sorry. Okay, so this last beaker then is isotonic. And that's when we have the perfect balance of inside the cell and outside the cell, the same amount of water. And again, why would that be? Because I would put solutes into this solution. We call that isotonic. The water is still going to move, but it's important, you know, the water molecules move one in the cell and one out. So they move in equilibrium. It's not that they don't move at all, but they move one for one. Better, worse?
Hi, Professor Stewart. What's the percentage for the isotonic solution?
So it would be the same. So if in my example, since I used 80% water inside the cell, I would put 80% in the solution. Yeah, I realize you can't see that very well, but I can do.
Okay, thanks.
All right. That's just, these are just for passive transport, right? For active transport, and we have all kinds of different things, right? So we have ion pumps, was what was discussed in your book and on the PowerPoints. And then also, I don't think we did, that's just for bacteria. So ion pumps, and then endocytosis, which has subcategories of pinocytosis, phagocytosis, receptor-mediated endocytosis, and then exocytosis. And all of those are active transport. They all require ATP because you're going against the concentration gradient. You're moving molecules from a low concentration to where there's already a higher concentration. Yes, Dan, what's up?
Could you repeat the percentage for the isotonic? You were kind of cutting out.
Sure, so the isotonic is the same. So it's 80% inside the cell and 80% of water outside the cell in my example. I kept it the same inside the cell for all of these cells. I would say 80% water, right? It's just randomly that I picked. Different cells at different times might have different concentrations of water in them, right? So like a bacterial cell might have 82%, or a red blood cell might have 90%, right? So we just kind of give it as a range. But in my example, I just used 80.
All right. Okay.
Awesome. Other questions, worries, concerns you have for chapter 7? Yes, no, maybe so. No, good.
Hypotonic and hypertonic. Could we like relate them to like hypo means slow and hyper means fast? Or, like,
Not necessarily. It's nothing to do with the speed.
Yeah.
So in this case, hypo always means over. You can think of it that way. Hypo refers to over, and it has letter O in it, right? So hypo, over, that's where the higher percentage of water is, if you want to think of it like that. But no, it has nothing to do with speed.
Yeah, okay. Thank you.
You're welcome. Anybody else? Yes, no? All right, so I'm gonna, if not, please ask me any questions you have about chapter 7 right now, because I'm gonna end this recording and start a fresh one for chapter 8 so that it's whole and complete and separate from chapter 7, and you don't have to try to fiddle through the recording if you're looking for a specific thing from chapter 8. I myself am just flipping through chapter 7 real quickly here to be sure that there isn't anything that I left out. My recollection is that there are 2 videos for chapter 7, a part 1 and a part 2. And the amount of detail that you need to know, for example, about the membrane is in the first video, is in part 1. All right. Great. All right, then I'm going to end this recording.